Hello everyone, my name is Salma Metwally. I'm a second-year medical student, and today I'll be explaining to you this lecture on digestion and assimilation. Okay, let's get started. So, in this lecture, we will talk about the digestion and absorption of four main things: carbohydrates, proteins, lipids, and at the end, some vitamins. Okay, but before we start with these, we will go over some general concepts that we should keep in mind throughout the lecture. Okay? So first thing, let's define absorption and digestion. So what is digestion? Digestion is basically breaking down the things that we eat by chemical processes into smaller molecules that can be absorbed by our intestinal lining, by our intestine. Okay, so the key in digestion is that it's a chemical process. And so for that, we will need digestive enzymes. These digestive enzymes are secreted in the saliva, in the gastric juices, and the pancreatic juices. Now, for absorption, after we have digested the food that we ate into these small nutrients and particles, we need to absorb them. So absorption is basically the movement of nutrients, water, and electrolytes from the lumen of the intestine across the intestinal lining into the basolateral side so that we can take them up into blood. Okay, and absorption can be via two pathways. First is the transcellular pathway, which is basically transporting them across the cells. And so for this, we use transporters on the apical side and on the basolateral side. Don't worry about these. We will see them again halfway through the lecture. But I just want you to know the general idea of transcellular absorption is through the cell. Paracellular pathway is the other way we can absorb these things, and basically they go between cells across these tight junctions that you have. Okay, and in general, transcellular is for mostly nutrients, while paracellular is for water and electrolytes because it's just easier for them to go that way. Okay. Now this is the lining of the intestine. This is the basic structure that you find on the intestinal lining. It's a villus. Okay, so the villus is this part that's going up. It's kind of like a fold of the intestinal lining, and then it dips down to form a crypt. Okay. Now, absorption mainly happens at the upper third of the villus, okay, by these absorptive enterocytes, the one we see in pink. Now, in the crypts, we have these stem cells, okay, these red ones. So stem cells basically are pluripotent cells, and what pluripotent means is that they can divide and mature and differentiate into different kinds of cells. So stem cells at the crypts divide and mature and differentiate, acquire their special enzymes and so on, and they move up the villus until they reach the tip. Okay? When they reach the tip of the villus, they will die by apoptosis and then they shed off into the lumen of the intestine. This process of dividing and migrating and then shedding off is called epithelial turnover. Okay? The idea here is that you're creating a balance between making cells and removing cells, and it takes around 3 to 6 days. Now we also have other kinds of cells here in the villus or in the intestinal lining. You have goblet cells, enteroendocrine cells, and Paneth cells, and you should probably know these from histology. Okay, now why do we have villi in the lining? It's to increase the surface area for absorption. Okay. Now digestive enzymes. So we know we have three main classes of nutrients: carbohydrates, proteins, and lipids. Now, another thing we should know is that digestion may take place at like any of these three locations, either in the lumen or at the brush border, which is basically the apical surface of the enterocytes, where you have these microvilli, or digestion can be intracellular inside the enterocytes. Okay, the nutrients, the three different kinds of nutrients get digested at different locations, but Generally, all three of them will be digested in the lumen, okay? And digestion in the lumen is usually by pancreatic enzymes. The other two locations, some of these nutrients will be digested there, some other ones will not be, but lumen is where you have all of them being digested, okay? We will see what we mean by this in a second, right? Now, you have these pancreatic enzymes, as we said, in the lumen, and basically you have different enzymes for different nutrients, okay? So carbohydrates, lipids, and proteins, they have different kinds of bonds in them. And so you have different enzymes to target these different bonds. For carbohydrates, the enzymes target glycosidic bonds. For lipids, they target ester bonds. And the enzymes for proteins will target peptide bonds. Okay? Okay, this is a flowchart that I made for you guys. Don't worry if it looks like a lot. But hopefully by the end of the lecture, inshallah, you will be, familiar with all of these and be able to identify them. So I just want you to look at this part. So nutrients, again, as we said, carbohydrates, proteins, lipids, okay? And as we said, they can be digested in different parts. So carbohydrates, for example, are digested at the lumen as well as the brush border, okay? Proteins are digested in the lumen, brush border, and intracellularly inside the enterocytes. Lipids are only digested in the lumen, okay? So again, all of them will be digested in the lumen, okay? And we have different enzymes for different locations and for the different nutrients. And we'll see these, throughout the lecture now. Okay, so this is our first checkpoint. We're done with the introductory part. Hopefully you're all caught up with that part so that we can move on to the actual bulk of the lecture, okay? So yeah, let's start with the first part, which is carbohydrates. So we'll start talking about carbohydrates and their digestion and absorption, okay? Now, first we need to know like a basic structure of carbohydrates. Carbohydrates are made up of these small units called monosaccharides. Then these monosaccharides join together to form oligosaccharides. And oligo basically means few. So oligosaccharides could be, 2 monosaccharides, it could be 3, and so on. So in this case, this, for example, is a disaccharide. Disaccharides are oligosaccharide, okay? And then when you have a lot of these monosaccharides joining together and to form like long chains or these branched structures, they're called polysaccharides or complex polymers, okay? Now, monosaccharides are glucose, galactose, and fructose. Oligosaccharides are maltose, maltotriose, sucrose, lactose, and trehalose. And the polymers are starch, cellulose, and glycogen. Okay. Starch can be found in two forms. It can be amylose and it can be amylopectin. Now, what is the difference between them? Amylose is a linear structure, while amylopectin is a branched structure. Why is this important? Because we will have different bonds, between them. So first of all, the most basic kind of bond that you would find in a carbohydrate is this alpha 1,4 linkage, okay? So this is the bond that will join disaccharides, it will join amylose, it will join the chains of amylopectin, but not the branches. So any linear linkage of monosaccharides is by alpha 1,4. Any branch is by alpha 1,6, okay? So for example, amylose is only made up of alpha 1,4 linkages between the monosaccharides, while amylopectin is made up of both alpha 1,4, which you would find here, for example, between these two, and alpha 1,6, which is found at the actual like branch, okay? So like here and here, okay? Then we have a third kind of bond, which is beta 1,4, and this is generally found in cellulose. And something about this type of bond is that we cannot break it down, and we will see why in a little bit, okay? Now, Oh, another important thing is that carbohydrates can only be absorbed as monosaccharides. So when you're digesting carbohydrates, you need them to become monosaccharides at the end so that you can actually absorb them and take them in. Okay? Carbohydrates, as we said, are digested in the lumen and at the brush border, but not inside the enterocytes. Okay? Yeah. So let's look at the actual digestion. So we'll start in the lumen. And the main enzyme that will digest carbohydrates in the lumen is alpha amylase. Okay? Alpha amylase basically breaks down the complex polysaccharides into oligosaccharides, okay? Not into monosaccharides. And why is that? Okay, let's look at it first and then we can figure out why. So amylose, for example, as we said, is a linear polysaccharide. It only has alpha-1,4 linkages. So like this bond over here, this bond, and even this bond, and all of these bonds are alpha-1,4, okay? Now, alpha amylase can only break down alpha-1,4 linkages that are on the inside. So it can like break down at this point, it can break down at this point, but it cannot break down this one over here. It cannot do that, okay? So it cannot break down the edge. And so for that reason, what we will end up with is something like this or something like this. We cannot break down this bond over here or this bond over here, and same goes for this bond. Why? Because they are on the edge, okay? You can only go on the interior side and break down those bonds, okay? And that's why alpha amylase gives us these oligosaccharides. This is a disaccharide, and yeah. So these are not monosaccharides. Now, another thing about alpha amylase is that it cannot break down the alpha-1,6 linkage. And where do we find this? We find it in branched complex polysaccharides like amylopectin. So this is an amylopectin over here. We have alpha-1,4 here, 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 and also here and here and so on. But this bond is an alpha-1,6 bond because it's a branch, okay? It's connecting these two chains together. So alpha amylase cannot break down this alpha-1,6 linkage. And so when you break down this amylopectin molecule, you end up with this maltotriose with maltose. Again, you can't make monosaccharides, you can't make glucose, for example, because you can't break down the edge. But because it's an amylopectin with an alpha-1,6 linkage, you also end up with something called alpha-limit dextrin. Alpha-limit dextrin is basically this, this molecule which has the alpha-1,6 remaining, okay? Because we couldn't break it down with alpha amylase, all right? So luminal digestion by alpha amylase, we end up with oligosaccharides, including this alpha-limit dextrin, maltotriose, and maltose. We don't have monosaccharides yet, all right? So this is basically what we said, complex polysaccharides into oligosaccharides. The main enzyme is alpha amylase, and it's an endoenzyme. What this means is that it breaks down the interior alpha-1,4 linkage. It cannot break down the terminal alpha-1,4, and so you don't form monosaccharides on their own. You cannot break down alpha-1,6 of amylopectin, and so you end up with alpha-limit dextrin. And another thing that is that you end up, you can't break down the beta-1,4 bond. Where did we say this was? In cellulose, right? This is why we cannot break down cellulose, because our enzyme alpha amylase cannot break down its bonds, okay? Okay, now we have alpha amylase from the saliva and from the pancreas. Which one is better or like more effective? It's the pancreatic one. Why is that? When you eat something, you don't keep it in your mouth all day. At some point, you have to swallow it, right? So this food, this like food that you ate, is not exposed to saliva for very long, okay? And so saliva, like the alpha amylase in saliva, cannot really do its effect, right? So that's the first thing. It's not exposed to the alpha amylase in saliva for long. Another thing is that when you swallow the food and you're swallowing the saliva with it, the saliva contains alpha amylase. This like food and amylase saliva mixture goes to the stomach, right? And what do you have in the stomach? You have acid, and you have proteases and all these enzymes that break down proteins. What is alpha amylase? It's an enzyme, which is a protein. So when it gets to the stomach, it would be broken down. So the one that it was in saliva reaches the stomach and gets broken down. Pancreatic alpha amylase, however, does not pass by the stomach because it will be released into the small intestine right away. So it doesn't get destroyed by the stomach acidity. And it will be exposed to the food for longer than, saliva. So this is why the pancreatic amylase is more effective. And you will see this generally with the other nutrients as well, that the pancreatic enzymes are generally like the most important, basically. Also, the pancreas releases a lot of alpha amylase, and so, it does the job well. Okay? Okay, now we're done with the luminal digestion. We have our oligosaccharides. We don't have monosaccharides yet. So we need to break down these oligosaccharides into monosaccharides. And the enzymes that will do this are the oligosaccharidases, okay? The name tells you what it does. So oligosaccharidase, ase is an enzyme. So when you add this to anything, it's enzyme that breaks. So this is an enzyme that breaks down oligosaccharides, okay? And this happens at the brush border, okay? So we said that carbohydrates get broken down in the lumen and at the brush border. Now, at the brush border, we have the oligosaccharidases, as we said, to break down oligosaccharides into monosaccharides. And we have four main oligosaccharidase enzymes. The first one is called maltase glucoamylase, okay? The name is kind of a mouthful, but, the first part of it tells you what it does. So maltase basically means that it breaks down maltose, right? So we know that we have maltose and maltotriose, which are oligosaccharides. Where did we get them? From breaking down amylose and amylopectin, okay? So when we break down these by maltase glucoamylase, we get glucose. Glucose is a monosaccharide. So we reached our goal, okay? Another oligosaccharidase is sucrase isomaltase, okay? This enzyme is actually a bit special because it's essentially two parts. So sucrase and isomaltase. Now, sucrase, from the name, you can tell that it will break down sucrose, right? When sucrase breaks down sucrose, it will convert it into glucose and fructose, both of which are monosaccharides. Okay, the next part is isomaltase. Now, isomaltase is very important. Why? Remember when we broke down amylopectin and we, ended up with the alpha limit dextrin, which had the alpha 1,6 that we couldn't break down with amylase, sorry. So isomaltase will do the job of breaking down the alpha 1,6 linkage that we couldn't break down initially. So it breaks down the alpha limit dextrin into maltose and maltotriose. All right, and these two can go to the maltase over here and be broken down into glucose to get our monosaccharides. All right, so if we go back to this picture over here, you see the alpha limit dextrin. So as we said, isomaltase comes and then it will break down this bond, which is the alpha 1,6. So we end up with this, which is basically this one, maltose, and we end up with this one, which is basically this one, maltotriose. All right. Yeah. The next enzyme that we have is lactase. Lactase breaks down lactose into glucose and galactose. Okay. And the last oligosaccharidase that we have is trihalase. Trihalase breaks down trihalose into two glucose molecules. All right, so glucose, fructose, and galactose are monosaccharides. Maltose, maltotriose, sucrose, lactose, and trihalose are oligosaccharides. Okay, so now we have our monosaccharides, and we can move on to absorption. But before we do that, let's look at this picture. So this kind of shows you a summary of everything we said so far. So again, starch, which is a complex polysaccharide. Amylase breaks it down into disaccharides or oligosaccharides in the lumen. Now, these disaccharides include lactose, sucrose, and maltose, which will be broken down by their respective enzymes into the different parts. And all of these at the bottom, glucose, galactose, and fructose are monosaccharides. Okay? Now, we said that we can only absorb monosaccharides. And now we have our monosaccharides. Again, glucose, galactose, and fructose. How do we absorb them? Okay, first to orient ourselves, this is an enterocyte. This is the intestinal lining. Here is the lumen. Um, this is where you basically have all the food going in at first. And on this side is basolateral, okay? Which is basically facing the interstitium and blood vessels, which will be, running over here, okay? So we're here and we want to move all the way to this side. How do we do that? Okay, we have channels or transporters, more correctly, transporters at the apical side. We also have other transporters on the basolateral side. We have two main kinds of transporters at the apical side, the SGLT1 and the GLUT5. SGLT1 is sodium glucose transporter 1. GLUT is basically glucose transporter, okay? Yeah. So SGLT1 is responsible for transporting glucose and galactose across the apical membrane into the enterocyte. And it will need 2 sodium molecules for that. So this is an active process. It uses sodium to, kind of get this energy and drive glucose and galactose into the cell, okay? GLUT5, however, is a passive process. It will transport fructose into the cell without using any energy. It's kind of just like, a channel for fructose to go through, okay? So SGLT1 is for glucose and galactose. GLUT5 is for fructose. You can kind of remember that 5 starts with an F and fructose starts with an F, so, they're kind of related. And SGLT1 is for these two because these two they look really similar and they are pretty similar. So they use their own like similar channel, okay? The same channel basically. Now we said that this needs sodium, right? So in order for sodium to go into the cell, we need to have very little sodium inside the cell. How do we maintain this low sodium here to have an electrochemical gradient for sodium? We need this famous sodium-potassium ATPase, or sodium-potassium pump. And it's usually found at the basolateral side. So what does it do? Basically, it will send sodium out of the cell and potassium into the cell. We don't need to worry about potassium right now. We're looking at sodium. So it pushes sodium out of the cell in order to, remove it from here and have low sodium here so that this can flow down its concentration gradient, okay? And when it does that, it will drag these two with it. Okay, now we're in the cell. We have our glucose and galactose, and we have our fructose, which are all monosaccharides. We need to take them across the basolateral side so that we can take them to blood, right? So that we can use them. Now, thankfully, they go through the same channel at the basolateral side. And this is GLUT2, okay? So glucose, galactose, and fructose, they go through GLUT2 across the basolateral side to go into the interstitium. All right, so you can remember that this is the first step. This is the second step. So GLUT2 for the second step, and GLUT5F for fructose, and then, SGLT1 for glucose and galactose. And you have to remember that this is SGLT1, okay? You have to know the number. Why? Because we have another channel called SGLT2, which is found in the kidney, okay? And so you have to know the different names, different numbers, all right? Now, we absorbed glucose, but not all carbohydrates, not all carbohydrates get digested in the small intestine. Some of them keep going until they reach the colon or the large intestine. When they reach there, they will be fermented by our bacteria in the colon, the microbiota, okay? Fermentation is basically an anaerobic process. It doesn't use oxygen, and it converts these carbohydrates into gases. So carbon dioxide, hydrogen, and so on. And these bacteria are mostly anaerobic bacteria, which is good because, fermentation is anaerobic, and so there's absence of oxygen. All right. Now, why is this something that we have to keep in mind? Because in someone who has lactose intolerance, they cannot digest lactose. So why? Because they have deficiency of this enzyme lactase. Remember, this is an oligosaccharide which would work at the brush border to break down lactose, okay? If they don't have it, they cannot break down lactose in the small intestine, and lactose just keeps going until it reaches the colon. And there it will be fermented by the bacteria and produce all these gases. Also produces lactate and acetate. So the gases will cause bloating, and because we have this lactose in the lumen still, it drags all the water to where it is and causes osmotic diarrhea. Okay? Now, to diagnose the lactose intolerance, there are two main methods. There's the hydrogen breath test and there's the lactose tolerance blood test. Hydrogen breath test, basically, you give the patient lactose, and if you measure hydrogen in their breath, it means that they most likely have lactose intolerance. Why? Because measuring hydrogen in the breath means that the lactose got fermented to produce this hydrogen. Okay? And if it got fermented, it means that, it didn't get digested properly in the small intestine. Another test is lactose tolerance blood test. Basically, what you do is you give the patient lactose, and then you measure their blood glucose. So what is lactose? It's a carbohydrate. It will give us, eventually it will give us, glucose, right? So when someone ingests lactose and, they digest this lactose and absorb the glucose and everything, their blood sugar or blood glucose should rise. Now, in patients with lactose intolerance, they cannot digest it. They cannot take glucose from it. So if they ingest lactose, their blood glucose will not rise. Okay? We reached another checkpoint. So I hope this part is clear. If it's not, you can go back to it and repeat it until it, becomes clear. Now we will answer a couple of questions. So since this is recorded, you can just pause the video and read the question, solve it on your own, and then you can come back and I'll discuss the question. Okay? So I'll give you like a few seconds to pause and so on. Okay, this is our first question. Okay. So this question asks us about a substance that needs to be further digested before it can be absorbed by the intestine. And the answer for this question is B. Okay, sucrose. Sucrose is a disaccharide. It's an oligosaccharide. And we know that we can only absorb monosaccharides. So sucrose must be digested further so that it can be absorbed. All right? Okay, next question. Okay. This question just wants us to pick the correct answer. So, okay, I hope you paused and answered the question on your own. Okay, so the answer for this question is D. Okay, the uptake of fructose into an intestinal epithelial cell is independent of sodium gradient. Okay, because we know the channel or the transporter for fructose is GLUT5. Okay, GLUT5 does not need sodium. It's independent of sodium. The other one, which is SGLT1, if we go back to this is GLUT5 for fructose, no sodium. SGLT1, which is for glucose and galactose, needs sodium. Okay, so this is our answer. The other statements are incorrect. Okay. Okay, so we finished the first part. Now we will move on to proteins. So now, okay, the proteins that we have in our small intestine for us to digest and absorb can either be exogenous or endogenous. When we say exogenous proteins, they are basically coming from the food that we eat. And when we say endogenous proteins, they are coming from, all the things that we secreted into the digestive tracts, all the digestive secretions. They have enzymes and they have all these proteins in them. We need to recycle those things. We can't just let them go to waste. So we need to break them down and absorb them. We also have proteins from the shed cells. Remember how we talked about the turnover of the epithelium. So shedding these cells releases all these proteins with them, and we need to reabsorb those back, okay? Like reabsorb the proteins so that we can, use them again. Okay, now proteins can be digested in the lumen, at the brush border, or inside the enterocytes, okay? When we talk about the brush border and intracellular digestion, we are mainly talking about peptidases, okay? When we talk about luminal digestion, we mainly talk about pancreatic proteases, okay? Again, as we said, pancreatic enzymes are the main ones, basically. So pancreatic proteases are two main groups, endopeptidases and exopeptidases. Endopeptidase basically is an enzyme that breaks down a peptide bond that's on the interior, okay? So it's kind of similar to alpha amylase, breaking down the bonds on the interior, remember that? So endopeptidases, they do that as well. Okay, they go to the inside of the peptide chain and they break down the inner bonds, okay? Some endopeptidases from pancreatic enzymes are trypsin, chymotrypsin, and elastase, okay? Then we have exopeptidases. Exopeptidases basically remove the amino acids from the edges, okay? So they kind of do the opposite of these. So they go to the edge and they chop off amino acids one at a time from the C-terminal end of the chain, of the peptide chain. And these exopeptidases are basically carboxypeptidase A and B, okay? We have something called enteropeptidase. This enteropeptidase is not the same as endopeptidase, okay? Be very careful about that. Enteropeptidase is also an enzyme that activates the endopeptidase trypsinogen into trypsin. Trypsin is the active form of this enzyme. When it's released by the pancreas, it's released as trypsinogen, which is an inactive form. So in order to activate it, we need enteropeptidase, okay? Now, another thing about proteins that makes them different from carbohydrates is that they can be absorbed as amino acids, but they can also be absorbed as di and tripeptides, unlike carbohydrates, which had to be monosaccharides. Here, proteins can be amino acids, they can be peptides, di and tripeptides. All right? Okay. Now, this is kind of the whole process of protein digestion and absorption. So let's go over it. Okay. We start with dietary proteins. These are the proteins that we ate in the food and so on. When they go into the stomach, they will be exposed to pepsin and gastric acid, which break down these proteins into smaller peptides. But these peptides are of variable lengths. They're not really readily absorbable yet. We need to actually digest them well in the small intestine. Okay, and we start in the lumen. So again, we said that we have the pancreatic proteases. They're divided into endopeptidase and exopeptidase. Again, trypsin, chymotrypsin, and elastase are the endopeptidases. Carboxypeptidases A and B are the exopeptidases. So we said exopeptidase chops off the amino acids from the edges. So they give you these amino acids on their own, which can just go on and be absorbed, okay? Endopeptidases, trypsin, chymotrypsin, elastase, they break down the peptide bonds that are on the interior. So when they break down the peptides, we end up with oligopeptides again. Kind of similar to what we had in the carbohydrates, okay? Now, these oligopeptides, if they're not di and tripeptides, they're still too long to be absorbed, okay? So we need to break them down into di and tripeptides or into amino acids, but at least they have to be these di and tripeptides. So we are done with the luminal digestion. Now we move on to the brush border digestion. The brush border basically takes these oligopeptides that we have here and it breaks them down into these smaller di and tripeptides and amino acids. And there are two main enzymes that are found on the brush border that will do this. And these are the oligopeptidases and the aminopeptidases. Okay? So now we have our di-tripeptides, and we have even more amino acids, and all of these can be, readily absorbed. Okay. Okay. So again, this is exactly what I was talking about. Yeah. Now, absorption of amino acid. So we have different transporters for amino acid absorption, but in general, the transporters for amino acids are not very specific. So any amino acid can be transported by any amino acid transporter. They're not really limited to their own transporter, okay? As long as it's an amino acid transporter, it can be, you know, like moved through it, okay? Now, at the apical side, we have this uptake of amino acids that uses sodium, okay? And what does that mean? It means that we need a sodium potassium ATPase on the basolateral side, okay? To keep the sodium inside the cell low and give us this electrochemical gradient so that we can take up amino acids, okay? If we go to this picture, you can see these channels, these transporters over here, taking up the amino acids, and they take sodium with them, all right? So sodium going in, amino acids going in. And again, we have the sodium potassium ATPase on this side, or the pump, in order to remove sodium and keep a gradient of low sodium inside the cell so that sodium can flow down its concentration gradient and drag amino acids. Now, once the amino acids are inside the enterocytes, they can either be used by the cells for different processes or transported across the basolateral membrane to go into blood, to be used by the rest of the body. Okay? Now, at the basolateral side, we have facilitated diffusion of amino acids without sodium. Okay? So we said that here we use sodium, which means that it's an active process. At the basolateral side, when amino acids cross, they don't use any sodium. They just flow by facilitated diffusion, which is a passive process. And then they can, go to blood and so on. Okay? Something that you should know is that you rarely have these diseases that are caused by a deficiency of an amino acid, due to defective amino acid transporters in the GI specifically, because, amino acids can just flow into any amino acid transporter, okay? So you barely have that. You do, however, have a disease that results from defective amino acid transporters in the kidney, okay? So normally when you lose amino acids into the urine, you need to absorb them back into the body. And if someone has defective transporters in the kidney, they can't absorb the amino acids back from the kidney. And so they stay in the urine and they basically form kidney stones. And this condition is called cystinuria. Okay? Now we absorb the amino acids. We still have some peptides to absorb. Remember, they are di and tripeptides. So we have a bunch of channels here. We still have our sodium potassium ATPase on the basolateral to, keep low sodium inside the cell. Okay? This time, we're gonna need this low sodium for a different channel, which is the sodium hydrogen exchanger, NHE. Basically, sodium flows in because we have very little sodium here. And when it flows in, it pushes hydrogen out into the lumen, okay? Here is the lumen, and here is the basolateral where you have blood, all right? So this NHE basically pushes out hydrogen. Why do we need that? Because the transporter that will take up the peptides is called PepT1, and this time it needs hydrogen instead of sodium. So it needs the flow of hydrogen into the cell so that hydrogen, flowing in can drag the di and tripeptides with it. Okay? So now you have the peptides in here, and you also have amino acids that we absorbed from, before. Okay? You can transport these di and tripeptides again by facilitated diffusion at the basolateral, just like we did with the amino acids, so that they can all go to blood and be used by the rest of the body. Or these di and tripeptides inside the cell can be digested into amino acids, and then the amino acids can just flow. This is done by peptidases. So remember we talked about the luminal digestion. We also talked about the brush border digestion. Luminal was by the pancreatic proteases, the endopeptidases, and the exopeptidases. The brush border was by these oligopeptidases and the aminopeptidases. And now we are talking about the intracellular digestion of proteins by peptidases. So breaking down these peptides into amino acids. And then all of these, the peptides and amino acids, go by facilitated diffusion across the basolateral side. Okay? Something that we should know about is called peptidomimetics. Now, this is, just know it very briefly. This has to do with pharmacology. So basically what this means is that there are some drugs which are resembling peptides, and those include antibiotics like beta-lactams and cephalosporins. These are some antibiotics, and they also include ACE inhibitors, okay? These drugs, they resemble peptides, so they can be absorbed by similar mechanisms. So when you take them, orally, and they go to your GI, they can just be absorbed by these similar mechanisms because essentially they are peptides, okay? And we reached another checkpoint. So now we will answer a question and then move on to lipid digestion and absorption. So you can go ahead and read the question, and then you can come back and we'll discuss. Okay, so this question basically asks about a drug that activates a proteolytic digestive enzyme. So proteolytic means it's an enzyme that breaks down proteins. And this enzyme is released as a zymogen in the pancreas. So it's released as an inactive form and needs to be activated by a certain substance, a certain drug. What can we use for this drug? The answer for this question is C, enteropeptidase. Remember, we said enteropeptidase is an enzyme that activates trypsinogen into trypsin. Okay, trypsinogen or trypsin is a proteolytic digestive enzyme. All right. And by the way, if you want to check the slides, the actual slides that I'm using, I've placed the answers and the explanations for these questions in the notes section, so you can do that. Okay, now let's talk about lipids. So, for lipid digestion, we have, it's only happening in the lumen, okay? And the main enzyme that we need is lipase. But we also have other enzymes like cholesterol ester hydrolase and phospholipase. Lipase is for triglycerides. Cholesterol ester hydrolase is for cholesterol esters, and phospholipase is for phospholipids. So all these break down their respective molecules. Okay, lipase can be gastric lipase, it can be pancreatic lipase. Like the other ones, the pancreatic lipase is generally the more important one, the more effective one, okay? Gastric lipase has a small effect, but it's pretty important in newborns and in pancreatic insufficiency in adults. So basically, newborns have defective pancreatic enzymes because, they're still not adapted to eating lipids and all that, okay? So they, instead of using pancreatic lipase, they break down their lipids using gastric lipase. In adults with pancreatic insufficiency, they don't really have like pancreatic lipases to use as they need them, so they use the gastric lipase, okay? Now, gastric lipase breaks down triglycerides into diglycerides and one free fatty acid. Pancreatic lipase breaks down triglyceride into monoglyceride and two free fatty acids. I forgot to add the two, but it's shown over here. So this is a pancreatic lipase, and this is a triglyceride. Here we have a glycerol in the middle, okay? And it's stuck to three fatty acids. So this is one, this is two, and this is three. What pancreatic lipase is We said that it gives us monoglyceride. So this over here is a monoglyceride. We see one glycerol with one single fatty acid, and it lost two free fatty acids. What the gastric lipase will do is it will only remove one fatty acid, and the other one will just remain here, and this will be called the diglyceride instead. Another difference between these two is that gastric lipases don't require bile salts, and this makes sense because, you don't have bile salts in the stomach, okay? But pancreatic lipases require bile salts, and they also require something else called colipase, and we'll see how this works in a few slides. So, another thing we need for lipid digestion in the small intestine, as we said, is bile salts. Bile salts are involved in two main processes, emulsification and solubilization, and those are two different things, okay? And we will get to know them in a little bit. Okay, so let's start with the first part of lipid digestion. First, we have emulsification and hydrolysis. Okay, now this is a fat globule, okay? This is a big chunk of lipid. You can't really digest this, efficiently, because if you go with the lipase at the surface and you try to break down these, you'll just take way too much time. So you need to increase the surface area by splitting these into into a lot of small droplets like these, okay? But you have another problem. When it comes to lipids, you know that they are hydrophobic. And the intestine environment is basically full of water, and, it's a very watery environment. So lipids, they can't really mix with water, right? So what they will do is they will keep clumping up into this big chunk of lipid. Even though the mechanical contractions of the intestine breaks down the fat globule into smaller lipid droplets, okay? You can't just rely on that, because as we said, these small droplets, even if you get them by the mechanical separation, they will still combine back together and go back to this big chunk. So you need to kind of form a barrier around them to prevent them from recombining with each other. And the thing that does that is bile salt. Okay, bile salt basically, as you know, is released by the liver, and it has two sides, two surfaces. One side is hydrophobic, and the other side is hydrophilic. Now, when they form this little like ball around the lipid, they keep their hydrophobic side on the inside, facing all these lipid droplets, and they keep the hydrophilic side on the outside, facing the environment inside the intestine. Okay, so this is emulsification. When they emulsify it, they kind of like keep them separated from each other and allow us to digest them efficiently. Okay, so now we separated these lipid globules into droplets, and we can start digesting, right? Well, not quite. So we have our enzyme lipase, okay, the pancreatic lipase. It comes, and then it can't really anchor itself and digest. Why? Because the bile salts form the barrier. So lipase needs some help, and it gets it from colipase. So colipase is a cofactor for lipase, okay? Colipase comes and attaches to this ball, okay? And this allows the lipase to get anchored on it and basically do its job, okay? So colipase attaches here, allows lipase to come and attach and then do its job. Colipase is actually secreted as procolipase, which is inactive, and again needs to be activated to become colipase. It's activated by trypsin. If you remember, trypsin is a pancreatic protease, okay? And procolipase is activated by trypsin to become colipase, and then go and help the lipase digest this, okay? Okay, so now we have digested all these lipid droplets in here, and we produce these lipid products, okay? But we can't let these lipid products just swim around in the intestinal water. right? Because again, they are hydrophobic. They can't do that. So we need to form another ball around them. And we will use the same thing. We will use bile salts again, but it's going to be a different kind of like ball, okay? So we're using the same component, bile salts, but we're getting a different collection. We're getting something called micelle. So this process is emulsification. In order to digest, this process is solubilization by the micelles, by bile salts forming micelles. And this part, the solubilization and forming micelles, basically is to gather all these lipid products that we got by digestion and be able to transport them to the actual surface of the intestinal lining, to the enterocytes, so that they can be absorbed. Okay? Now, we have the micelle carrying all our products that we got from digestion, and we need to absorb them. Now, this is where lipids are different from the carbohydrates and proteins. So carbohydrates and proteins are water soluble, right? So that's why they couldn't diffuse across the membrane on their own. They needed some transporters to help them. Lipids, however, are lipid soluble, right? The membrane is lipid, so they can just diffuse by simple diffusion. And that's exactly what they do, okay? So they enter by simple diffusion. Inside the enterocyte, they will be re-esterified using free fatty acids. Esterification basically means adding fatty acids to these lipid molecules. Yeah. Now, after we esterify them, they will be placed into something called chylomicron. Now, be careful about these three, this emulsification droplet, this micelle, and this chylomicron. They're all different, okay? The emulsification droplets and the micelle are both produced by bile salts. Chylomicron is not produced by bile salts, okay? We're in the enterocyte. We're inside the actual cell. So chylomicrons are produced by phospholipids, and they're also produced by something called apolipoprotein B48, okay? So the way you can remember this number, B48, so 48, 8 from eating. So this is for all these lipids that you ate, basically. Okay, so B48 for the chylomicron, all right? Now we form this chylomicron, which has all these lipid things that we broke down and absorbed. And then chylomicron moves here into the lymph. Then from the lymph, it can go to blood. Why can't it go to blood right away? Because it's too big, essentially, and we need it to go to the lymph first so that it can actually go to blood, okay? And lastly, what happens to the bile salts? They stay in the lumen. They keep moving until they reach the distal ileum, and then at the distal ileum, they get absorbed and they will get recycled back to the liver, okay? So that we can use them again, all right? Okay, this is the flow chart that I showed you guys at the very beginning. So I hope it makes sense now. So let's go over it really quickly again. So carbohydrates, we said lumen and brush border. At the lumen, the enzyme is alpha amylase. And then at the brush border, we have oligosaccharidases, which include maltase glucoamylase, sucrase isomaltase, lactase, and trihalase. Okay. Now, for proteins, we have lumen, brush border, and intracellular digestion. At the lumen, we use pancreatic proteases divided into endopeptidases and exopeptidases. Endopeptidases include trypsin, chymotrypsin, and elastase, which are pancreatic, and the stomach endopeptidase is pepsin. And these are the ones we talked about because these are the ones found in the small intestine, not pepsin, okay? For exopeptidases are basically carboxypeptidases A and B, okay? Then for the brush border, we have peptidases, and those are oligopeptidases and aminopeptidases. And for intracellular, we have peptidases. Now for lipids, in the lumen, we have lipase, bile salts, and colipase. Okay, so we reached another checkpoint, and we are almost done. We just have a little bit more. We have a question coming up, so you can pause, solve it, and then come back to discuss. Okay, so this question asks you about, it's a very simple question, asks about something that is made by the liver to aid in fat metabolism. And the answer to this question is A. Okay, bile salts. Bile salts are made by the liver and they help in fat metabolism. Be careful about chylomicrons. Chylomicrons are not made in the liver. They are made inside the enterocyte. Basically, when you collect all these lipids that you broke down, you form a chylomicron in the intestine, in the enterocyte of the intestine, so that it can, be absorbed into the lymph and then into blood. Okay? Okay. Now, the last part of today's lecture is talking about vitamins. And we have two main vitamins to talk about. We have vitamin B12 and vitamin B9 or folate. Okay? So first, let's talk about B12. B12, another name for it is cobalamin. Um, so when you eat something that has B12, it's basically stuck to a protein, okay? So this red thing is B12, and this yellow thing is a dietary protein. So when you eat it, you will swallow it, but you swallow something else with it. You have this thing called haptocorrin, which is secreted by the salivary gland, okay? But it doesn't do anything here yet. We will see what it does in a little bit. So when you swallow the B12 stuck to a protein, and you swallow the haptocorrin. They go to the stomach together. Remember what we have in the stomach? We have acid and pepsin. They break down this protein that is stuck to the B12. So the B12 now is free, but we need something to, stick to it. So the haptocorrin that we had from the saliva will stick to the B12 here in the stomach and, continue with it for a little bit more. We have something produced by the parietal cells in the stomach called intrinsic factor. This is very important, and we will see why in a little bit. So intrinsic factor is made by the parietal cells in the stomach, and then it will, flow with this B12 haptocorrin complex to go to the small intestine. Now, inside the small intestine, B12 will be freed from the haptocorrin, okay? And now, again, it's free. So what it does is it joins this intrinsic factor, okay? The B12 intrinsic factor complex over here will be absorbed in the distal ileum by endocytosis. So this is the ileum mucosal cell. This is a cell in the ileum. The B12 intrinsic factor complex goes there and gets absorbed. Inside, it will be freed from the intrinsic factor in the lysosome. Okay, this is a lysosome. We remove the intrinsic factor and B12 is now free. So we need to transport B12 through the blood so that we can actually use it, right? We need to send it to the liver so that it can do all of its functions and stuff. So B12, in order to be transported in blood, it needs to be combined with something called transcobalamin, okay? Cobalamin, it comes from cobalamin, which is B12. So yeah, so B12 joins with transcobalamin, and they go into the blood and basically delivered to the liver so that we can, have it used by all these functions. You can ignore this side of the picture. This just shows you some processes done by B12. It's completely irrelevant to our topic today. But yeah. So basically you can see this is a B12 with the transcobalamin, and it goes to a cell and does its function, okay? Now, we can have vitamin B12 deficiency, right? And vitamin B12 deficiency causes a specific kind of anemia called pernicious anemia if it's due to a lack of intrinsic factor. So how do you get vitamin B12 deficiency? It can be, due to a certain diet or certain like way that somebody eats, if they restrict meats and all that, they get vitamin B12 deficiency. But another way somebody can have B12 deficiency if they don't have intrinsic factor. And in this case, lack of intrinsic factor, it would be pernicious anemia. Why would they not have intrinsic factor? Where was it made initially? It was made in the parietal cells of the stomach, right? Parietal cells. Now, if someone has an autoimmune disorder that destroys these parietal cells, they can't make intrinsic factor and they cannot absorb vitamin B12 efficiently, and so they get a deficiency of vitamin B12. Or if somebody has done a gastrectomy where they remove the stomach or part of the stomach, basically they have less of these parietal cells, they have less intrinsic factor, less absorption of B12 and deficiency. How do you treat B12 deficiency? You just give supplemental vitamin B12. All right. And lastly, we have folate, also called vitamin B9. Now, folate or folic acid is another name for it. Has a very fancy name. It's teroyl monoglutamate. Yeah, this is another name for folic acid, and it can also be found as a polyglutamate. Okay, so when we eat certain foods that have folic acid, that have polyglutamate, we need to break them down into monoglutamate, which is the folic acid. And so to break down the polyglutamate into monoglutamate, we use the enzyme folate hydrolase. Okay, so folate hydrolase converts this polyglutamate into monoglutamate. When you get this monoglutamate or the folic acid, you can actually transport it into the enterocyte now. And the transporter that does that is PCFT, proton coupled folate transporter. So it's over here. So it transports folate or the monoglutamate into the enterocyte, and it's coupled to proton or hydrogen. So it needs hydrogen to flow with it. Okay. So this is similar to the mechanism that we saw with the peptide absorption, right? The PEPT1, okay? So we saw this NHE or sodium hydrogen exchanger when we were absorbing peptides. It's a similar thing. So we have hydrogen going in with the folic acid, and in order to have this, we need this NHE exchanger, which exchanges sodium for hydrogen, and for sodium to flow in to get hydrogen out so that it can go with folic acid. We need low sodium here, and we have the famous sodium-potassium pump at the basolateral to do the job, okay? So now we absorb folic acid, and it can be used by the body for different functions. It's important for DNA synthesis. So if somebody has a folic acid deficiency or a folate deficiency, they have compromised DNA synthesis, and this leads to macrocytic anemia. And these patients basically require dietary changes and supplements, okay? So another thing is that serum folate is actually high in SIBO. SIBO is small intestinal bacterial overgrowth. So basically they have a lot of bacteria in their small intestine where, we shouldn't have all these bacteria in the small intestine. But in these people who have a lot of bacteria, basically these bacteria start to make vitamin K and B9. Okay, B9 again is folic acid, right? So a lot of bacteria in the small intestine, they start to make vitamin B9 or folic acid, and that's why they have high serum folic acid levels. All right. And now we have a question, and this is the last part. So we can go ahead and read it. Okay. So this question talks about this man who has an immunologic disease, and this disease has destroyed his parietal cells of the stomach. What would be expected for this patient? So the answer for this question is E, pernicious anemia. So This man has an immunologic disease, and basically it destroyed the parietal cells of the stomach. We know that parietal cells make intrinsic factor, which can be used for the absorption of B12. If we destroy these parietal cells, we can't make intrinsic factor, and we can't absorb B12. So this is pernicious anemia, the lack of intrinsic factor, and leading to B12 deficiency. All right. This is the end of the lecture. I hope you benefited from it. And if you have any questions, feel free to contact me. I left my phone number and my email at the very beginning, so you can go ahead and send me a message if you have any questions. I would be glad to answer you. And please don't forget to scan the QR code to evaluate the session and help me improve in the future. Thank you so much for watching, and good luck.